Hey, I just wanted to quickly plug that if you guys want any tutoring, some new slots have opened. If you look in the description, there's a link to my physics and maths tutor profile in which I do tutoring on there. Okay, welcome back to another episode of the A-Level Cookbook. Today we're going to be doing the AQA A-Level Biology Paper 3 from June 2017. So it says, when a nerve impulse arrives at a synapse, it causes the release of a neurotransmitter from vesicles in the presynaptic knob. Describe how. So a synapse is a connection between a nerve and another nerve, or it could be a muscle, a gland, or any other sort of effector like that. And the synapse is the connection between these two and how they interact, and the synaptic cleft is this gap here. What happens is all depolarizations in A-level biology are due to sodium channel ions coming in. So an action potential will be here, right? We'll already have that sodium coming in, sodium ions. So then what happens is we need to somehow get something across here to then essentially trigger an action potential here. What happens is, because there's depolarization in this presynaptic neuron, what happens is we get an influx of calcium 2 plus ions now because of the depolarization. So V-gated calcium ion channels open. Then what happens is we have these vesicles called neurotransmitters that fuse here like that. They will release neurotransmitters, like these are chemicals that will diffuse across and bind to a complementary receptor like that. When they do, it's going to cause depolarization in here. So if we're getting depolarization, sodium ions have to come in. So what happens is you get ligand-gated Na plus ion channels opening, Na plus enters, and you get a generator potential here. If you hit the threshold, if you got enough of this neurotransmitter binding on this, then bang, you've got an action potential like that. Once the job's done and dusted, this acetylcholine or whatever it is, the neurotransmitter, is broken down, removed and taken back in, and these calcium ion channels are also shoved back out as well, because we don't want to just keep having it bind to a receptor like that. So here, all this question wants you to say is to describe how it's getting your action potential releasing neurotransmitter. That is all. They don't want anything else beyond that point. I was just saying how synapses work entirely. So what happens is that depolarization, because the question wants it from step by step, from impulse to there. So depolarization causes voltage-gated Ca2 plus ion channels. You can't just say calcium, you have to say calcium ions, or you can just say Ca2 plus channels to open. So then Ca2 plus enters via facilitated diffusion, right? And then what happens is it causes vesicles containing the neurotransmitter to fuse with the cell surface membrane. What this does is this releases the neurotransmitter into the synaptic cleft. And there you go. That's all the steps they want, which is the gap. They don't want you to go and talk about like the rest of everything else. All they want you to do is just describe those steps to there. So just be cautious when you're reading questions like that. The presynaptic knob contains actin, filaments, and myosin molecules. The myosin molecules can attach to mitochondria and move them towards the presynaptic membrane as shown here. In this diagram, we can see a mitochondrion. There's a myosin fixed to it, and for some reason that can make it move this way towards the presynaptic membrane, and we have an actin filament. It says, use your knowledge of how actin and myosin interact to suggest how the myosin molecule moves this mitochondrion towards the presynaptic membrane. It says, do not talk about calcium ions and tropomyosin in your answer. I'm just going to talk to you about how they just generally work to see what the crack is about. So here, normally what happens is you've got your myosin head chilling, right? And it's not able to bind to actin just yet. I'm not talking about how I'm answering this, I'm just giving the background knowledge. Normally what you have is you've got tropomyosin and troponin here. Calcium ion channels cause this tropomyosin and troponin to change shape, dragging it out the way, making that binding site on the actin nice and free. So what happens is this myosin head binds here and ATP is hydrolyzed and it moves this way, hinges, like swings that way, right? And drags the actin that way. That's what happens. Now, it says use your knowledge. Now, your knowledge says that actin only binds to myosin only binds to actin, right, and drags it across. So we need to think about using this diagram, how that would explain that. We can't say that the myosin's binding to here. That's not right. So what's happening is that the myosin will bind to the actin and do its power stroke. So it will swing this way, right? And as a result, if it's swinging that way, then, like this whole thing kind of rotates that way, and this is going to go that. Can you see how it's here? Here, it's going to drag this across like that, and this like that. So as it's swinging, the mitochondrion is being pushed that way, if that makes sense, moving it along the actin. So the actin is just doing its normal job. So what you would say is that this myosin binds to actin and it hinges, it bends, it whatever's you want to call it, you can say power stroke, and then this moves the mitochondria along the actin, right? Because again, if it does that hinge, it's just going to move that way because this actin's fixed there and it's just chilling. So there you go. So the movement of mitochondria happens when nerve impulses arrive at the synapse. Suggest and explain one advantage of the movement of mitochondria towards the presynaptic membrane when the nerve impulses arrive at the synapse. So we have to think of what the possible uses are in regards to mitochondria and synapse. Mitochondria make ATP, which supplies energy. So you need to think, at synapses, when would I need energy? Now, when the neurotransmitter is moving across, it's doing it just standard diffusion, right? It's happy in days. But when we're taking it out, right, we're going to have to actively transport it back into the presynaptic membrane so that we can actually, get them back in, right, as quick as possible. And also, we need to remake it and regenerate it. So, for example, acetylcholine esterase will break down acetylcholine, its products are taken back, and then we're going to have to remake it, and that takes energy. So the use of it there would be something like that. So the mitochondria produce ATP, and this could provide energy for active transport and reuptake of neurotransmitters. Or you could say you're remaking them as well. That also works. There you go. So bacteriophages are viruses that kill bacteria. Figure 2 shows drawings of a bacteriophage and a bacterium. Using figure 2 in your own knowledge, put a tick in the box next to the only correct statement about the structures of the bacteriophage and the bacterium. So here it says both have ribosomes, right? This has told you it's a virus. Viruses don't have ribosomes. They only just contain genetic material in a capsid, that's all. So immediately that one's out. Both have a cell surface membrane, same thing. Viruses do not have a cell surface membrane, so that's out because this has told you it's a virus and it's your own knowledge. The bacteriophage has a capsid and the bacterium has a cell surface membrane. Well, the bacterium definitely does, and viruses are just capsids with like DNA material and stuff in it and other bits and bobs, right? So that's definitely that. And then the bacteriophage has a cell wall and the bacterium has a capsid. That's not correct because viruses don't have a cell wall. So the only one that is correct is this one. There you go. So using the scales in figure 2, calculate how many times larger the bacterium is in the bacteriophage. Use the distance between the points labeled A and B in your calculations and show you're working. Okay, so I can't really measure this. I've tried using the ruler, it just doesn't work. So what you need to do is you've got to work out the distance here to here in centimeters, work out the magnification here, distance here to here in centimeters, work out the magnification here, and then work out the actual the image size, and then use the magnifications to find the actual sizes, and then you would do actual bacterium, is that what it is? Yep, divided by the actual phage. Like that. An alternate route is you can measure this in centimeters and say, well, this many centimeters is this many nanometers. So then this many centimeters would be X nanometers. And then the same for this. Compare the units, make sure they're the same and so forth. I can't do that just because I've tried with the ruler, it just don't work. Scientists investigated the use of bacteriophages to treat lung infections caused by bacteria. What they did is they infected the lungs of mice with the pathogenic species of bacterium, and the mice were then divided into two groups, A and B. So A was not treated. B was treated by breathing in a spray that had these particles. After 3 days, what they did is they killed the mice, removed their lungs, washed out the lungs with a set volume of liquid, and then they determined the number of bacteria in the liquid. Then here's their results. It shows the mean and the range of the data about the mean. Standard deviations are not shown. Fine. So first of all, let's have a look at what their independent and their dependent is, right? So it's going to be the independent is phage spray versus none, right? And the dependent, what they're measuring is the number of pathogenic bacteria in fluid from washing the lungs, right? This might be useful. Now here it's showing us the data, it's showing us the mean and the range, and these aren't standard deviations because the standard deviation will plot the spread of data around a mean. So you'll have your mean in the middle of it. These are ranges because you can see this is the highest value, that's where the mean was, and that's the lowest. So just be careful because there have been people that have been like, well, the standard deviations don't overlap, blah, blah, blah. Like that's not the standard deviation. So if we have a look at the data just real quick, without being treated, they have a higher mean amount of live pathogenic bacteria in this wishy-washy fluid that they got versus in B, where they had a lower amount. Now, the range of values don't overlap, but that doesn't give us any help with like statistical significance and all that jazz. We can't say for sure that they're statistically different because we don't have a p-value to test it. So that's something that's interesting to just keep in mind. So anyway, it says, what would their null hypothesis be for this investigation? So because we're looking at whether we're looking at phage spray versus no phage spray, and we're looking at the number of bacteria. The null hypothesis would be that the bacteriophage, makes no difference. Whoops, the pen's in the way. Hold on, Ish. Makes no difference in the number of living pathogenic bacteria. Because we're looking here, our hypothesis is, oh, well, there might be a difference if we give them bacteriophages. So the null hypothesis is that there's going to be no difference between the two of them. And the only reason I can tell is because I've just measured, written out the dependent and independent variables. The dependence, the mean number, and we're doing this spray versus no spray, then obviously we're looking to see if there's a difference. So a null hypothesis is that there's no difference. With some samples, the scientists decided that they needed to carry out a series of dilutions in the sample before counting the bacteria. Use evidence from figure 3 to explain why dilutions were necessary for some but not others. So if you have a look at figure 3, right, these are all singular numbers. It's the log 10 value, right? To undo a log 10 value, you need to do the base, which is 10 to the power of the log 10 thing. So for example, 2, right? 10 squared, that's 100. 10 to the 9, is 10 followed by 0 9s. Can you see how the range of values is gigantic, right? And if you have a look, we're going from what, 10 to the 6 points, like what, 6.6 to 10, 10 to the 8.8, which is huge range compared to here, which is like what, a couple tens to nearly a couple millions, right? So the range of values is gigantic, but the question's asking you, based on that, why would they do dilutions for some but others? If I tried counting colonies in a plate and it's all just this, I can't count them, right? So if I dilute them, then I'm going to spread them out so I can actually count them a little bit more easily, and then I can undo the dilutions to eventually figure out what was the original thing here. So that's exactly what it is that the first thing is that the logarithmic axis, the log values, show a very large range of results in the number of bacteria, right? So then dilutions are necessary for high counts or high values to be able to actually count them, to be able to count the number of bacteria colonies or just bacteria. Because again, if you had what is that? Like 9 billion of them, there is no way in hell you're sitting there counting 9 billion. It's just not practical. So now it says, what can you conclude from this data about the effectiveness of the phage in treating lung infection in mice? Do not consider statistical analyses in your answers. And here you can't talk about overlaps and this and that because there is no SD here. And they've also instructed you not to. So here then, what it's saying is, can you say this is effective at treating this in this? So what we're doing here then is we're looking for supportive statements and then contradictory statements. All right. As well as that, we can talk about validity as well. But here it's just saying, is it effective? Yes or no. So what we're going to do is we're going to look at what's supportive and what's going against it. Now, if we have a look, without the bacteriophage, the mean is higher, whereas in the bacteriophage, the mean is lower. So immediately you can say that the bacteriophage reduces, the number of pathogenic bacteria, right? And this is in all mice of each group, of group B. Because can you see how there's no range overlap? None of these have the same amounts of these. So clearly the lowest tier was different to the highest tier, sorry. So that means there's no overlap because no overlap. That doesn't mean they're not significantly different, blah, blah, blah. It's because we don't have a statistical test. But we can say in this one experiment with all of these results, these lot had no overlap. So in all cases, they were lower here versus here. That's all we can say, but we don't know whether it's statistically significant or not. However, we're looking for contradictory statements as well. So what's the problems of trying to say, oh, this is reliable as treatment? Well, I mean, it's not gone to zero immediately. That doesn't treat it, doesn't cure it. So it doesn't reduce to zero. But also, we have a huge range of results. Can you not see that there's this there's still loads left here versus 10? So there's a huge range of bacteria as well. Some are like tens, others are nearly millions. You don't really want to give a treatment kills such a huge range. And some people it cures it, and other people it's you've got a couple bajillion bacteria left, no big deal. And there you go. That's how I'd tackle this. We don't really need to talk about we don't know what species and this and that, blah, 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 because the question's asking you directly, how useful is using bacteriophages in treating lung infection in mice? So we're not going to be it's only been done in mice, not humans, because that's not what they're asking you to do, bro. Farmers use artificial fertilizers to maintain or increase the yield of grain-producing croplands such as wheat. Artificial fertilizer is used to replace mineral ions removed from the land when crops are harvested. One of these is nitrate. Give 2 examples of biological molecules that contain nitrogen that's removed when you're harvesting a crop. So here, people were just saying nitrates, ammonium, ammonia. That's not what it's asking. It's saying 2 examples of biological molecules. Biological molecules are your amino acids, your carbohydrates, your DNA, RNA, ATP, proteins, all of that jazz, right? Well, so here, we're not going to really say carbohydrates because they don't have nitrates in them. So it's going to be any one of the other ones that have nitrates in it. So for example, amino acids, or you could say DNA. The reason it's not ammonia, nitrates, ammonium, urea, and all those jazz is because those are inorganic things. They're not biological molecules. Well, urea is a waste thing that we get rid of as well. So we're not really taking that out, and that's made in from the deamination of proteins in the liver and then gets peed out. So scientists investigated changes in the use of artificial fertilizer in India between these 2 years. They also investigated, they also investigated change in the fertilizer response ratio. This ratio shows how many kilograms of grain are produced for each kilogram of fertilizer used. So please, for God's sake, write down what that means or something, because it's going to have that ratio somewhere here and people are going to fumble on it, and I can already tell. The fertilizer response ratio says kilogram of grain made or produced, or whatever you want to call it, over a kilogram of fertilizer used. Interpret that. What does that mean? So if you have a really high fertilizer response ratio, that's great because it means you're using very little of this and you're getting loads of that out. Make sense of the results. Don't just dive into the question and just try and hope for the best. Then it says, figure 4 shows the results in the form scientists presented them. A hectare is a unit of area commonly used in agriculture. So let's have a look what's going on here. This axis is showing the kilogram of artificial fertilizer per year that's used. And it's showing you the bars of the fertilizer response ratio on which this axis is there. So the kilograms of artificial fertilizer used is this line, right? So just make sense of it one by one, first of all. This is as time goes on, what's happening? The fertilizer response ratio is dropping as time goes on, generally. Then, separately, look at this one next. As time goes on, they're using more artificial fertilizer instead, right? Then do between the both of them. So as time goes on, the fertilizer response ratio goes down, but while that's going down, this artificial fertilizer is going up. So if you do it like that, then you've pretty much gotten the bag in terms of what you're going to go and then describe later on and explaining and evaluating and all that jazz, right? Which lo and behold is coming up. So then it says, uses data to calculate the difference in the mass of grain produced per hectare in 1970 to 2005. So get the numbers that you're going to need. So 2005, 1970, right? And it's saying the grain produced per hectare, right? So it wants the difference in the mass of grain produced per hectare in this compared to this, right? So we need to work out the values of each bit and bob. So if I do 1970 here, 2005 here, the axes are telling us the kilograms of fertilizer that we used and the fertilizer response ratio. But the question wants how much we made, how much grain. The ratio shows how many grain of kilogram are produced. You know how I wrote that there? That's what they want you to find. So we're going to have to record these fertilizer response ratios first, and then the kilograms of fertilizer used. So for 1970, let's get the fertilizer response ratios, right? So the fertilizer response ratio, these bars, and the axis is here, right? So if we look at 1960, the fertilizer response ratio was going to be what? So that's 1, 2, 4, 6, 8, no, so it's 4, 8, 12, 60, 20, right? So that means it's going to be 12.2. Then, if you look at 2005, it's going to be 3.8. Cool. And then we look for the kilograms of the fertilizer that was used. So in 1970, we're looking at this line here, and this is this axis here, right? So that's going to be what? That's 10, 12, 14. So there was 14 kilograms used here, right? And up here, it's going to be what? 60, 70, 70, 72, 74. Cool. So then if we want, remember the fertilizer response ratio is the kilograms of grains produced over the kilograms of fertilizer used. So the amount that we produced is going to be these two times together. So if I do 12.2 times 14, I get 170.8. And then if I do 3.8 times 74, we get 281.2. And then we want the difference. It's 281.2 minus 170.8. We should get 110.4. There you go. So then it says, use this data to evaluate the use of artificial fertilizers on grain-producing crops in India. So, I mean, what we can see here is, again, we've got to look at supportive and contradicting statements and validity problems. Now here, I've just said that the response ratio is dropping, which means we're making less kilogram of grain, but we're actually using way more fertilizer, and the data also shows that. So clearly, it's not looking too good. So what's happening is over time, more fertilizer is being used. That's what I said when I was describing it to begin with, but the response is decreasing. The idea is that it's over time, right? Here, there's not really a supportive statement, is there? I mean, it's saying, is artificial fertilizer good on these grain crops? I mean, it's entirely just a negative finding that you're using more and less is coming out. However, we still need to talk about our validity issues. So can I apply this to every single plant in the universe? Can I say this is 100% true? Is this answering the original research question? The original research question was in changes in the use of artificial fertilizer and changes in the fertilizer response ratio, right? But just because we're using more fertilizer and the numbers go, the response is going down, that doesn't really mean 100% that's the sole and entire reason only, right? There's a lot of other factors that come into growing crops. So correlation may be present, but correlation doesn't mean causation in this sense, right? It's not direct. There could be other factors, and you should always say what the other factors are. Don't really just say other factors. It doesn't really help, right? So there could be other factors, such as some mark schemes allow other factors, others don't. So that's why I just say don't. Just if you're going to say other factors, follow it up. Such as, like, for example, I don't know, soil quality, right? If you've got really rubbish soil quality and you're shoving it with fertilizer, there's no point. Nothing's going to grow. So ecologists investigated changes in the grassland communities of large islands off the coast of Scotland between these 2-year time periods. On each island, they used the data of the number of sites to determine the change in the mean species richness and the mean index and change in the mean index of diversity. And here it shows the plant species recorded at one site in one island. And it says calculate the index of diversity using this formula. So that's the index of diversity. N is the number of all individuals, the sum of them all, right? Sigma means the sum of, and it's going to be the number of each species. So the little n is the number of each species. So what we need to do is we're going to work out big N. Big N immediately is just all of those added up, right? So if I add up all of those, 3, 19, and so forth, we get 3 plus 19 plus 3 plus 3 plus 10 plus 16 plus 6, we get 60, right? So here, this is going to be a pain in the butt to calculate, but what you can just do is, this is n, right? So just add an extra column, n minus 1. So you would just do 3 times 2, for example, and 19 times 18, and so forth. So if you do all of that, I get 3 times 2 is 6, 19 times 18 is 342. 3 times 2 is 6, and then it's going to be 10 times 9 is 90. 16 times 15 is 240, and then 6 times 5 is 30. Right, so now I've got all of these, right? But it's sigma and n minus 1. So sigma, all of these is the sum of all of those. Sigma means sum of. So you're just going to do 6 plus all, 3, 4, 2 plus 6 and so forth. So 6 plus 3, 4, 2 plus 6 plus 6 plus 90 plus 2, 40 plus 30 gives you 720, right? So then the index of diversity is just going to be 60, big N, n minus 1, all over. Well, we just worked out sigma n minus 1, which is just 720. If we do that, we get 4.917, right? But we want to keep to the same number of significant figures as a question, so we'll just go with 4.92. There you go. So outline a method that ecologists could have used to determine the plant species richness at one site. Species richness is the number of different species. It's not to do with the number of within, all right? It's the number of different species. So for example, if I have this, and like this, and then some more of the triangles and circles and stuff. Species richness is we have square, so that's 1. We have triangles, that's 2. We have circles, that's 3. And we have an asterisk, so that's 4. Species richness is just the number of different species, right? So that's what we're going to work out. So whenever you're doing a method, just always helpful to identify your independent and dependents first. Like literally just scribble it out if you want to. So your independent variable and your dependent. Right, so the independent variable is we're looking at the plant species at one site. So we're sticking to the same area. It's just going to be the area within the one site we're changing, aren't we? Right? And dependent is the number of different species seen. So now you should think in your head, how are you going to measure that? Right? We have one big area. We've got all these plants all over the place and different things, whatever. We want to look at the different species. So what you do is you would just put quadrats in. So now we need to think about ways to control and make sure we're not doing anything too mucky and making sure everything's fair and valid. So the first thing is we put this into an area like a grid like this and we choose coordinates because then obviously no one's like, oh, I like to look at that area. I'll do that to get rid of bias. So what you do is you divide an area into coordinates and then you use a random number generator to generate coordinates to sample. Right? The reason for this is because this will avoid bias. Then what we're going to do is we're going to just basically look to see if there's any there. So what we do is we use quadrats. So you use many quadrats, or a large number of them, and count the number of different species in each quadrat. There you go. That's it. Because all we're looking for is, again, just those different species in each of those quadrats. That is all. So some ecologist results are shown in Table 2. They carried out a statistical test to find out whether any differences between these means were significant. So it's literally just told you. The p-values, so the values for p that they obtained are also in this. So whenever you're doing the statistical test stuff, right, loads of people love just saying the results are significant, blah, 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 and they just say a whole load of model answer memorized garbage. It don't work, and it makes like, every time, just think of it this way, like every time you do it, a Victorian kid is going to die. So don't do that. Look at whenever you have a p-value, actually define what that p-value is testing. It's told you in the question what it's testing, but sometimes it won't. If it's a t-test, the p-value is testing the probability of the difference in means being due to a chance. T-test. If it's a chi-square test, it's the p-value is testing the difference in the observed versus expected. And if it's a correlation coefficient, then it's going to look for the p-value is testing the correlation between the two, whether it's positive or negative. You've got to say the difference in the means correlation, whatever it is significant. So here the p-value is testing, it's told you here, the differences in means between 1975 and 2010. That's all it's testing. You identify what it's testing, then it gets quite easy to say this. So it says here, do these data show that there were any significant different changes in the grassland communities on these islands? Give reasons for your answers. So we have a look. We have an island here. We've got Islay, whatever, and we're looking at the change in the mean species. So if it's a plus, it means the species richness has gone up, right? It's told you that this p-value in the species richness between 1975 and 2010 is less than 0.001. So that's a significant difference, as in there's significantly higher. It's not due to chance. The difference is not due to chance. The change is not due to chance. So here then it says the change in mean index of diversity between this is plus 0.22, which means it's implied it's gone high. However, this is greater than 0.5, so this is not significant. So although there's a significantly higher, mean species richness in 2010 than there was in 1975, the index of diversity didn't change significantly. Here in columns A, it's gone up, and this is significant, whereas the index of diversity has also gone up, and this one is significant because that's less than a 0.01, less than or equal to, so that's significant as well. Here, this has gone down, and that's significant, but then here, this index of biodiversity has also gone down, and it's more significant too. It's significant as well, sorry. So then, for each one of them, that's what you're going to say. So group them if you can. So Islay and columns A, had significant increases in species richness. Collins A had a significant increase in the, I'm just going to write D for short, index of diversity, but not Islay. And then Harris had a significant decrease in both species richness and the index diversity, right? We need to actually say what this significancy bit means. It means that there is a greater than 5% probability that, and don't just say the result, the difference in, because it's the index of diversity, in D, between those two periods, whatever it was 19, differences between 1975 and 2010, there's a greater than 5% probability that they were due to chance. The differences between these. Can you see how I'm saying what the p-value is testing? I'm not just saying the results, or I'm just, you can't just say that. You have to be like specific and related to this example here. So name two enzymes involved in the semi-conservative replication of DNA. So semi-conservative replication, so you have two strands, you break them into templates, right? And these are held together by hydrogen bonds, remember? So you're breaking those hydrogen bonds. You got those two templates. DNA nucleotides will line up like this via complementary base pairing by forming hydrogen bonds like this. And then they're joined together by creating new phosphodiester bonds to these adjacent ones by DNA polymerase. They do not get joined like this. If you joined them like, with DNA polymerase this way, you would literally just have chunks of two like this and not a strand. So don't do that. So that means in order to have broken those hydrogen bonds to begin with, you need DNA helicase. It's in the name, helixase. Helixase doesn't sound right. So helicase, it breaks the helix by breaking those hydrogen bonds. And then we also have DNA polymerase to actually join those nucleotides together again. There you go. So sometimes damage occurs during DNA replication. One enzyme involved in repairing damage to DNA is ATR. ATR works as follows. What it does is it phosphorylates other enzymes in repairing DNA. It also phosphorylates substrates required to repair DNA. So then when ATR phosphorylates other enzymes, they become more able to bind to these substrates. Use your knowledge of enzymes to suggest why. So a lot of people, unsurprisingly, were talking about saying competitive and non-competitive inhibitor. And it's just like, I can just tell you're doing model answers here. It don't work. The question has told you that phosphorylating is adding a phosphate, right? It's told you that it's phosphorylating enzymes and they become able to bind to their substrates. Normally, an enzyme binds to its substrate because it is complementary, but this question's told you that for some reason, sticking a phosphate on it, right, then makes it able to bind to a substrate. So what must have happened is that this phosphate changes the shape of the active site to make it become complementary. It's no inhibition here. So what it does is it changes the active site shape by changing the tertiary structure, because that's what determines it. You can't just say 3D. Loads of people just love saying 3D. It doesn't mean anything. It's changing the tertiary structure, right, to make the enzyme active site complementary to the substrate. It's told you that they are like, this phosphorylation is making it better at doing the job, so it's becoming more complementary. That is it. There's no inhibition here. So then it says the enzyme catalyzed reactions activated by ATR only work if the substrates have been phosphorylated. Use your knowledge of energy changes in the enzyme catalyzed reactions to suggest why. So here then, one of the things that we do with phosphorylation is that we make things more reactive. So that's immediately one of them, is that it makes substrates more reactive. But we're not done really, because we need to say how. I mean, actually, to be fair, it says it allows in the mark scheme, but the way that it does this is because it reduces the activation energy needed for them to react. It's not an enzyme there, it's just sticking a phosphate does that. That's how it works. Sometimes a mutagenic agent causes DNA to break. A different enzyme called ATM binds to broken DNA. This leads to the activation of a protein coded for by a tumor suppressor gene. So the effect of ATM binding is to stop all cell division until this DNA is repaired. The mutation could result in the person having a non-functional form of the gene that produces ATM. What can you predict about the possible effects of having a non-functional form of ATM? So in this question, only 47% got 3 marks, because a lot of people didn't just follow the story of the stem and then actually use what the question told them. They were just saying a whole load of things, a lot of random bits and bobs. All of these questions tell you a story and they say explain why. So go through it bit by bit if you need to. So here, DNA breaks, ATM binds, and then what happens? It activates a protein coded for by a tumor suppressor gene. So this tumor suppressor gene is expressed, right? And then cell division stops. The question says, what can you predict about the possible effects of having a non-functional form of ATM, as in, this don't work no more? What could possibly happen? Well, DNA breaks, ATM doesn't bind to it anymore, no tumor suppressor gene expression, cell division will carry on, which means you could then end up having, unfunctional cells that aren't working and doing the job properly. That's all it's saying. It's not going to talk about cancer. There's no tumor bit here. It's just saying what effect you might have if this is the case. And that's exactly what it is. It's just applying the story. So then ATM does not bind to DNA. So the DNA is not repaired. Cell division continues. And, the daughter cells have damaged DNA. Oh, and the reason cell division continues, is because DNA is not repaired. Cell division continues because the tumor suppressor gene, is not expressed, right? And there you go. You could talk about increasing the risk of like tumors and stuff as well, but the question's not really told you immediately that cancer's happening as a result, really. So talk about the steps here, if that makes sense. But we're also not done because it's just saying it's a non-functional form of it. Well, remember that like there are diploid organisms and they can have 2 copies as well, so it may have no effect if diploid, but 2 copies could lead to no ATM working, and that can lead to problems like, your like cancers and stuff like that. So I guess you could say that's your effect and stuff as well there. So a student used the pitometer to measure the movement of water through the chute of a plant. The pitometer is shown in figure 5. As water is lost from the chute, it's replaced from water in the capillary tube. Water is evaporating out of the leaf. That process is called transpiration. Transpiration is not really like that's a mysterious thing that happens. No one can really define what it means. It means that water evaporates out of the leaf through the stomata. As a result, then what happens is because the water is cohesive means sticks together, the water evaporating out of here creates a tension and it pulls up this column of water. Imagine I have a string, right, in a tube like this. If I yank on this string, the string goes up, doesn't it? It's the same thing here. That's why it's called cohesion tension, because the evaporation of water from transpiration is giving a pull. It's pulling the string, right? So it's causing a tension. Tension is pulling. It's putting a tension on this column of water. So the entire column of water is dragged up. And the reason is because they show cohesion because of their hydrogen bonds. I mean, they stick together. That's what the cohesion tension theory is. It's driven by transpiration, but it's not only transpiration that's the piece of the puzzle in it. Transpiration is simply just water being lost from the leaf via evaporation. So anyway, in one experiment, the air bubble moved this much in 15 minutes. The diameter of that says calculate the rate of water uptake and give your answer in millimeters cubed per hour. So remember that this is a tube, right? And it's got a diameter of 1 millimeter, and it's moved a distance of 7.5 millimeters, right? So the area of a cylinder or any sort of cylindrical prism-shaped objects is the cross-sectional area times its length. So for example, if there's a triangle-shaped one, it would be the area of this times its length. So here, we need to find out the volume of this water that's moved, because all the water that's moved is the volume of water that's taken up. So the volume is going to be the area, cross-sectional area, so pi times the radius squared. The diameter of this thing is 1, so the radius is 0.5 millimeters, right, squared, times the length, 7.5. If we do that, we get a volume of 5.89, right? But this was in 15 minutes. So if 15 minutes meant that this volume of water was taken up, right? Then 1 hour, 60 minutes, would be 4 times as much, because 60 is 4 times 15, so then this would be times by 4. So if you times this by 4, we end up getting 23.56, which is roughly 23.6. There you go. Done. So the student wanted to determine the rate of water loss per millimeter squared of the surface area of leaf in this of the chute in this figure. It says outline a method they could use to find this rate. You should assume that all water loss from the chute is from the leaves. So we're looking at the rate of water loss per millimeter squared, right? So you need to measure 1, you need to measure the water loss, right? It's per millimeter squared. So 2, you need to work out the surface area of the leaf. And 3, this is the rate. So we need to work out the time frame, all 3 of these things. So then what you're going to do is you're going to figure out how we're going to work out, for example, the water loss, which obviously is using the pitometer volume. So we've done that part in millimeters cubed. We need to work out the surface area of a leaf. So one thing you could do is you could put it on square graph paper, draw its outline, and then count the squares, then times by 2 for both sides of the leaf, right? Because we're assuming that it's going to be like, above and below that it's lost. And then, the time frame, you just measure with a stopwatch, right? To work out the rate, then, you're going to do, it's the volume divided by, and you would say this, the volume of the water that's moved from before, divided by the surface area for each leaf, divided by the time. Because then that would give you millimeters cubed per millimeters squared area per time in minutes or whatever it was, or hours. There you go. So the rate of water movement through the chute in a pitometer may not be the same as the rate of water movement through the chute of an entire plant. Suggest one reason why. If we think of the journey the water's taken here, right? It's going into here and it's going into a plant. So when we're doing this, what we're doing is we're cutting a chute like this at a slant and letting it go straight in there. The question's asking you, why might not it be the same as like a real plant? Well, in real plants, the water enters the plant via the roots, right? So then here you go. So just one reason why is that the plant has roots, which means there might be a longer time for the water to move through the entire plant. I'm not too sure why there's a huge space here, because it's one mark, and I just find that really odd. But there you go. So aquaporins are a type of channel protein that allows the diffusion of water across membranes. One type of aquaporin called PIP1 can also transport carbon dioxide molecules across this membrane. So then it says figure 6 shows the structure of a water molecule and carbon dioxide, and they're drawn to the same scale. Suggest 2 reasons why water molecules and carbon dioxide molecules can both pass through PIP1. Right, well, remember that PIP1, it's told you, is a channel protein. A channel protein, so here's your phospholipid bilayer, right? Chilling. A channel protein literally just makes a hole like this for something to move through, okay? So if these are both able to move through this, then clearly they must be able to actually fit, right? So they're similar in size, therefore they can fit. As well as that, what else have we got going on here? Remember that these proteins work by things interacting with them. They need to shape, they need to be like complementary, they need to either fit or bind, right, for these to work. So in order for these to actually get through this, it's not like they can just freely float through. They also have to be the right shape. They need to be complementary. So here, the other reason must be is that because something common between them must be it. So they both have oxygen, so that's probably the bit that's complementary, right? So they both have, or you can just say they're both complementary. So therefore, they're similar in shape. So they're both complementary to the aquaporin. So I'm just going to write AQP for short. That's how they work. Otherwise, if it was just as simple as a hole here, then every single thing in the universe could just move right through it, and it kind of defeats the purpose. These channel proteins only work on specific things, and the reason it's specific has to do with shapes and complementary things. The entirety of biology and specificity is, most really just that. The scientists first produced transgenic popular trees. These trees all had a length of foreign DNA inserted, and this led to the production of a single-stranded RNA that specifically inhibited the expression of the gene for PIP1. The scientists then measured the difference in the amounts of PIP1 in the leaves of transgenic populars and in the leaves of wild type without the foreign DNA. The amount in PIP1 in the transgenic populars is approximately 15% than in the wild type. Using this information, what can you conclude about the effect of the foreign DNA in the transgenic popular trees? So there's a lot going on here, but just scribble down the stories side by side, right? So we've got the transgenic PIP1 trees, and then the non, right? So these have the DNA that's foreign, and that leads to single-stranded RNA, and that leads to inhibition of PIP1. These do not. And then for some reason, these geezers have 15% or 85% less PIP1, right? So here then, we need to conclude the effect of having this foreign DNA. Well, first of all, we can say that we need to figure out how this single-stranded RNA works. Single-stranded RNA then inhibits the expression of it. If we're dealing with single-stranded RNA and it's stopping the expression of this gene, every single gene is expressed by first being transcribed into mRNA and then translated. So if it's somewhere along the line, this is getting screwed up, or also here with transcription factory bits, right? So if we're dealing with single-stranded RNA, the thing that's coming into mind is interfering RNA. So this single-stranded RNA must screw up here. DNA's got its 2 pairs, it's not going to do anything, right? But with mRNA, that's a single strand. So what happens is this single-stranded RNA is actually complementary to the mRNA. What it will do is it will form complementary base pairs, and as a result, it will lead to the destruction of that mRNA. That's how it works. So then the single-stranded RNA forms complementary base pairs with the PIP1 mRNA, and that leads to destruction of the mRNA. So there's no translation, so therefore less PIP1 is produced, right? But then it says the effect of this. So this is just only the steps. We need to think about the consequences as a result, right? Now, because this is all to do with aquaporins helping the uptake of water, then that must mean this can lead to decreased H2O available for whatever process you want that uses water. So available for photosynthesis. That's the most obvious one. There you go. So you've got to follow it step by step. And remember, this whole topic with gene tech and all of this jazz is mucking about with transcription and translation. You don't express a gene unless you transcribe and translate it. So the transgenic populace still produce some PIP1, suggest why. So again, if you're producing PIP1, it means you must have transcribed it into mRNA and you must have translated it into this PIP1. So clearly, if they're still producing some of this, there's still some mRNA, so there's still some PIP1. So the only reason this could be the case is that not all mRNA was destroyed by the single-stranded RNA binding to it. That's the only reason that would make sense. I find this interesting because in this question's examiner report, it says, many students does not grasp the link between an ssRNA coded for the foreign gene and blocking of translation by interference with this mRNA. 63% of people got 0, which is kind of interesting, I guess. But I guess it's because they're saying it was because this was the first year of this new curriculum, so they might not have been as more familiar. But then it says in the next part, in this question here, given the failure of many students to comprehend what was required in this previous part, it was not surprising that only 28% of these people got the mark. Many were awarded a mark for suggesting that not all of the cells had been transformed as well. So that's a possible thing here, but it's just working with what the question's telling you. The scientists investigate the importance of PIP1 in the movement of water and CO2 through the tissues and leaves of popular trees. They measure the mean rates of movement of CO2 and water through the tissues and leaves of transgenic poplars and through those of wild-type poplars, and here's their results. So real quick, independent and are dependent. So what we're changing, what we're measuring. So what we're changing, number one, is obviously transgenic versus wild-type. And what we're measuring, CO2 movement and H2O movement, right? Now what we need to do is we need to look at these results one by one. So start with the carbon dioxide ones and then the water ones between the two of them. Or start by transgenic first or this. Just compare one for one, right? So here, this axis is the mean movement of CO2. This is the mean axis of mean movement of water. So if I do, for example, if I do CO2 for CO2, the transgenic ones have a lower overall mean rate of movement of CO2 than the wild type. Then if I move on to water, the transgenic had a lower mean rate of movement of water than in the wild type. Then you can compare like betweens. So in transgenic, they had less CO2 movement than water, whereas in the wild type, they were equal. And not even just that, we can go further and look into it more details. So here we can see that this is a standard deviation because your mean is there. It's the spread of the results around the mean. Here you can see these are showing the spread of the results around the mean. So even though it doesn't outright say this SDs, these are the SDs because it's the mean here and the results are spread. So you can see there's no difference in the overlap. There's no overlap in these two, so they're significantly different. Whereas here, these do overlap, so there is no significant difference here, right? So that means that there's no difference in the mean rate of movement of CO2 and water in the means of them in the wild type, but in transgenic, the mean rate of movement of this water is significantly higher than in the transgenic, the mean. That's what the significance is testing, right? So then we've now just done all those comparisons. We can now use that to think about the next question. So always do that. Now it says using only this figure, evaluate the importance of PIP1 in the movement of CO2 and water through the leaves of poplar trees. So it's saying PIP1 is important for this: supportive, contradictive, and validity problems. So supportive, contradicting, and validity. So supportive statements is what's directly supporting this, right? Results that say, yes, this is the case. Contradicting is what's directly going against it, right? What's saying nope. And validity is the problems with it. So can I reply, is it answering the actual question? Can I apply it to everyone in the universe? How can I repeat this? Is it reproducible? Those kinds of things. If you've seen my other videos, you will have heard me yap about it for a long time about the chocolate shitting example. Just go have a skim through for one of those. So then here in this figure, what's supporting this statement that PIP1 is important? Well, these transgenic ones don't make PIP1 anymore, remember? So a loss of PIP1 reduces CO2 and H2O movement. And can you see how these SDs, right? Like, they're not overlapping with these results. These results are not overlapping. So the SDs don't overlap. So they're significantly lower, right? However, any contradicting statements? Not really, because this is a pretty supportive thing to show that PIP1's importance, obviously going down. But when we come to things with like validity and issues like that, or just other weird quirks, right? You can see that there's a bigger effect on the carbon dioxide than there is on water for some reasons. There's a bigger effect on CO2 versus water movement. And also it's not zero either. So that means this could suggest not all movement of CO2 and water is through PIP1. So an improvement to this would to go and look at other proteins as well and all the other things that they do as well. 18% of people got only 3 marks and 10% got nothing. So that's reassuring that only 10% didn't get anything. So now for the essay, it says write an essay on one of these topics: the importance of nitrogen-containing substances in biological systems and the importance of diffusion in organisms. Now, I ain't gonna lie. If you're choosing the nitrogen one, you are a very interesting person, but whatever, you do you. So then, just as a report from this exam thing here, and I've seen this a lot of people, what they try to do is like, they just kind of don't really, either they try to go above the A-level spec and say something that's just either at GCSE level or like wrong. In one series, enough people had mentioned key stage 2 level stuff about the water cycle that it had to be mentioned in the exam report. Remember that this is about 20,000, 30,000 people, right? So key stage 2, as a reminder, is year 3 to 6, which is 8 years old to 11. We're not going to be writing 8-year-old answers in your exams, right? Don't try and just cheat the system. It just doesn't work, right? Now, the other issue as well, and it's quite interesting, is the people that went for, obviously more people went for the importance of diffusion. That's the nicer one, right? But many of the better essays were actually on the nitrogen-containing substances, presumably just because it's a harder essay. Here, as well, Tim, another important thing is it's about what you're bringing into this as well. In the mark scheme, if you end up saying, irrelevant stuff, you're locking yourself out bands because it's marked as 1 to 5, 6 to 10, 11 to 15, 16 to 20, and then 21 to 25. So let's say you write a pretty decent essay, all of it's good, all of it's pretty decent, but you've just screwed up in one topic and it's that one topic screwed. You can get up to 16 to 20 marks, right? But then if you try to act like Captain Big Nuts and you try and say something beyond the spec and that thing is also horrendously wrong, now you've just put in 2 irrelevant topics or incorrect topics, which has now locked you in 11 to 15 marks, even though everything else is perfect. So rather than taking the chance, in my personal opinion, I think it's best to write about 4 relevant, juicy topics that are on spec. Because if you do those perfectly, then you can get up to 20. The last 5 are not worth it given the risk of that happening. Because then you've just locked yourself out of what? So you've lost the last 5 and now you've lost another 5 and you've locked yourself out of 10 marks easy. So just don't do that. I don't think. It's not worth it unless you really, really know. And the other problem is a lot of people wrote massive amounts of detail of recall, just writing an entire story and then at the end say, this is important because without this we'll die. That doesn't count as importance. The idea is it has to be synoptic. You've got to show a link. You've got to relate and integrate everything by showing your application across the board. As in, here's this thing, this is important because without this, this could happen and this and that, right? For example, a problem would be like a lot of people were saying a gas exchange in the lungs, right? So they'd be like, oh, without gas exchange, we'd die. Well, that's great and all. But like, you've got to show that it needs to be in the right standard. If we can't do gas exchange, you can't get oxygen, which means if we can't do that, we can't have oxygen being a terminal or fine electron acceptor in oxidative phosphorylation, meaning we can't make ATP, and ATP is important for all important functions. Can you see how those links are made across the board? So you really need to be crystal clear on those sort of links across the spec. You can't just yap about one thing and say, but this will die. That doesn't help, right? Now, I'm going to talk about the diffusion one, but you can obviously do the other as well. The strategy I recommend is if you can try and spend about 5 minutes really just skimming through everything you know in your head, one by one, really just knuckling down on the topics you want to do. The rest, 25-ish minutes or 30, you can write the essay. Obviously, don't spend like ages on it, like 45 or whatever. But if you can try and train yourself to get used to quickly shoving out a plan in like 5 minutes max, and I mean plan by literally listing the points you want to say, then writing in 25 minutes, if you can, you'll be safe, kind of. Obviously, that's just, that's quite a tough boundary in time-wise, I know, and I appreciate that. So obviously not everyone can do that, but that's what I personally did myself. But then again, I write and think and speak really fast, so you'll have to adjust it to your thing. Now, what I mean by that is let's list out the importance of each of these topics. So think between all of the topics and pinpoint the exact thing. Don't just write the whole topic. Don't just write biological molecules. That's useless, right? We are going to really hone in on what the thing we're looking for is. The importance of diffusion. In topic 1, that's all to do with like biological molecules, right? So your proteins, enzymes, lipids, sugars, and all of that jazz. However, that doesn't seem very exciting, but water is pretty important because, water is the solid in which things are moving. So you could talk about water being the sort of means of diffusion and stuff like that, but it's not the strongest topic, so I'll move to something else. Topic 2 is all about cell membranes, cell structure and division, and then the immune system and all of that jazz. Cell membranes is pretty fat, is it not? Because if we look at cell membranes, it's the importance of diffusions. So if we need to think of examples of where this is relevant, right? So for example, it would be the movement of O2 and CO2 at the lungs, the alveoli of the lungs, for example, and also things like the capillaries as well in tissue fluid. Can you see how there's a link there made? You need to think of a use. Why would it be useful? Well, diffusions, there's that, right? As well as that in cell membranes, we can also talk about, your transport proteins and why they're useful because they help in facilitated diffusion. So transport proteins, think of a use. So you're like, in my head, where do I know any uses of transport protein, right? There's transport proteins, there's aquaporins, for example, in your collecting ducts. And that's important for water regulation. And then there's also GLUT4 for insulin, because insulin binds to them and it causes these channel proteins in the skeletal and liver cells to be fused in the membrane and make them more permeable to glucose so they can take them up. They're pretty important, right? So those are some decent examples. Then we've got the immune system. The immune system, not really huge amounts of diffusion or active transport to talk about really there. So moving on to topic 3 then. Topic 3 is literally all about like exchange and transport systems, like your lungs, your gas exchange services and all of the animals. So immediately one thing you can talk about is gas exchange services, right? And like the process of that happening, why that's important, as well as that mass transport, right? Because we rely on like the facilitated diffusion of the plants really, the sucrose getting into the sink cells, which can then be used in like respiration, for example, or stored as starch. So that's quite important because without it, they can't use the glucose in respiration to generate ATP, which is important to, you can't undergo glycolysis so that you can't make ATP. And it's important for like everything. And you also can't form glycosidic bonds with the monomers of it and stuff and store it as starch, for example, like breaking it into, its alpha glucose and whatnot. That, right? That's a pretty important link. Topic 4 then is all about DNA and protein synthesis. I mean, if we have a think, I'm not thinking of anything too hefty there, nothing that's like screaming diffusion, right? Photosynthesis and respiration, anything there? Well, in photosynthesis, we need the diffusion of CO2 through the stomata and the leaves, which, is for that. And we also need water. But the problem is that during transpiration, you get diffusion of water vapor out of the leaf, and that can actually inhibit photosynthesis and cause a problem. So you can see there's a balancing act there. As well as in respiration, you need a diffusion of oxygen, and that's all linked to your gas exchange. Can you see how the links are made? Right, there's that. Topic 6, like the entire thing, to do with nerves is diffusion. Facilitated diffusion of sodium ion channels is the reason why we get action potentials and generator potentials and all that stuff as well. So Na plus and, all of its action potentials and this and that, and also your K plus as well links with that. So, Na plus in, K plus out, all of that jazz. That's important for like, your receptors and your control of stuff, right? Control of heart rate and things like that. And actually sensing things. Synapses as well. Synapses, when you get to a nerve to a muscle, how do you actually make the muscle contract? As acetylcholine will diffuse across the synaptic left bind to receptors. So that's pretty important. So synapses are pretty hefty. As well as that, like we were saying here, the control of glucose with the insulin binding and then the facilitate diffusion of glucose in. And then as well as that, the control of blood water potential as well. So can you see how those links come together? So then, if I verbally describe like an example, we could say something like, diffusion is important in the control of blood water potential. For instance, if the low blood water potential is, low, osmoreceptors detect this, they send impulses to the pituitary gland. And what it will do is it will release antidiuretic hormone. It will move through the blood, bind to receptors on cells in the collecting duct. And what will happen is vesicles containing aquaporins will fuse, and this will increase the permeability of those collecting ducts to water, meaning that more water will move back out of the collecting ducts into the medulla of the kidney and be reabsorbed rather than being passed in the urine via facilitated diffusion. This is important because it regulates water potential in the blood and can increase it when it's too low, or on the converse, by reducing the amount of aquaporins, make it, put more urine out by decreasing the blood water potential. This is important because it prevents cells from shriveling and shrinking and, making sure that they actually can continue their functions. And also water is important in a lot of chemical processes like condensation reactions as well. So make sure we have enough of that. Can you see the importance part there? You describe the steps hinging on that one little bit. You don't just monologue about the whole thing. You just come down to that one bit where it's crucial. So for example, in gas exchange, you could say something like, for example, all gas exchange surfaces rely on diffusion because they do. For example, in insects, the tracheoles, O2 diffuses out into cells and then CO2 diffuses back in to tracheoles and then they're, shoved out by the spiracles and arrhythmic abdominal movements. In fish gills, O2 diffuses out of the water into the gills, CO2 out the gills into the water. In lungs, O2 out the alveoli into capillaries, CO2 out into the alveoli and then breathe out. So can you see how all of those hinge on diffusion? That's pretty important because if we, we also have things to maximize that, like your high surface area volume ratio, thin diffusion pathway, blah, blah, blah. The reason this is important is because effective removal of carbon dioxide means that it won't dissolve in the blood and make it more acidic, which could cause proteins to denature and not function anymore. But also on the flip side, we also need to make sure we have enough oxygen, because if we don't have enough oxygen, we can't use enough of it in oxidative phosphorylation to accept the vinyl electron, and then we can't make as much ATP, and ATP is required for all living processes. Can you see how you're making those links? You've really got to hone in on it and say, this is this thing, this is why it's important. Without this thing, this other thing around here would be screwed up, and there, step by step. You're showing links between each topic. Not just, without this thing, we would die. That's not useful. Right, cool. Anyway, I hope that helps. Please like, share, and subscribe. You know the drill. And yeah, thank you very much.